on board. So welcome aboard. And Brian is now, this is taking slides to a whole new level, okay? Okay, so Brian is going to tell us about the omics revolution.
Okay, thanks, Dr. Lynch. So I've been here about 4 months, and it's been a great experience so far. I have great collaborators, many of who are in this room. But I'm going to tell you kind of about where we're heading in kidney cancer. We're a little bit behind a lot of our other solid tumors, but we're moving forward. And with the advent of the TCGA, which is becoming moving forward in kidney cancer, a lot of exciting things are on the horizon. So I'm just going to tell you where we've been, where we're going, and what we're doing at Yale. So again, we're just going to talk about, first we're going to talk about really classification schemes and how this also relates into how we end up treating patients down the road, understanding the biology of the tumor, really what tools are available, and again, what the TCGA has started to tell us. And I'll give you some initial information from 2 of the working groups which aren't published yet. I just got underway, and then also what we're doing here. So really, we're a little behind in kidney cancer. You know, we have changed the name, we have changed techniques, you know, but we've basically relied on light microscopy to really evaluate with kidney tumors. At least we don't say a hypernephroma or Grawitz tumor anymore, but having looked at pathology reports sometimes in the community, you still see these terms. But we've kind of split and lumped things into different categories. But as of 2012, we were still calling things, you know, clear cell, did have papillary morphology, and greater than 75% of the features, it was a papillary tumor. But we really don't have really good classification schemes in terms of the genetic basis or analysis. But lo and behold, this year we came up with a new classification scheme based on the Vancouver meeting, and only 2 of these classes actually have a genetic kind of basis for their evaluation. Most of them are still morphologically or immunohistochemically kind of subclassified, and that is actually important when we start moving towards molecular classifications to actually target cancers. We look at some of our breast cancer colleagues, and they're way ahead of us. I mean, right now, you know, 75% of tumors are clear cell, and I'll get in. You know, clear cell is very heterogeneous. Papillary is very heterogeneous. But you know, we're a little bit behind, but hopefully we'll catch up with the advent of the TCGA. In 2003, this is a slide from my mentor while I was at the NCI, Marston Linehan, who described some of these genes, VHL and MET in kidney cancer, and also discovered the folliculin gene. In 2003, this came out, the genetic basis of kidney cancer. I think this was a big, you know, really exciting. You know, urologists at least said, oh, we discovered it. Our medical oncology colleagues also thought, you know, now we know what these morphologic subtypes mean. But this is really based on the study of hereditary kidney cancer. Just because there's a hereditary link between sporadic clear cell and VHL-associated clear cell doesn't mean that many of these syndromes share the same genetic aberration as some of the hereditary syndromes, such as HLRCC, hereditary leiomyomatosis renal cell, or hereditary papillary renal cell, where they're predisposed to getting certain types of kidney tumors. And then chromophobe and oncocytomas are present in something called Burt-Hogg-Dubé. So I think this was exciting, but I think it also may have held the field back because a lot of people tried to link the hereditary and the sporadic forms together. But now with all these tools that are available, things are drastically changing. You know, IHC, Cytogenetics have been available, but we're starting to move towards, you know, the genomics era. And just looking where that term genomics came from, actually, there's some basis from Yale. One of the scientists here actually who studied Hox genes actually was on a journal which they actually called Genomics. And that basically began the genomics era, where we're actually moving towards a lab-on-a-chip techniques where we'll be able to further kind of characterize some of these kidney cancers. You know, we all know they come from the nephrons. Did they come from the proximal part? Did they come from the distal part? You know, now we're actually able to really interrogate it in much more detail. And, you know, as many of you are familiar, you know, you can do all these different tools to study cancer. You have, you know, you could study expression with just a gene chip, an Affymetrix chip. You know, you could do RNA-seq. You can have look at copy number variation, or you can do you can do next-gen sequencing. So all these tools are available for us to study kidney cancer, and we know it's a really heterogeneous class of cancers. when you're talking about, kidney cancer, and you're trying to characterize it by mutations and you try to sequence it, kidney cancer, you see the clear papillary and chromophobe. There's a lot of mutational events, and really trying to do targeted therapy for some of these cancers where you have multiple mutations. Some of them are drivers, some of them are passengers. It's really hard to kind of discover really what are the actionable items in some of these cancers which are highly mutated. But the TCGA is upon us. Kidney cancer, the clear cell paper has come out in the past year. But what is the TCGA for people who don't know? It's a $400 million kind of the war on cancer, as Harold Varma said. It's a collaboration between the NCI and the NHGRI, and really we're doing molecular characterization of a lot of these different cancers. Kidney was lucky to have papillary and clear cell chosen, but as part of the Rare Tumor Initiative, chromophobe was one of actually the first projects to be added in, and that's the third most common type of kidney cancer. It has the potential to identify new possible targets, and then we can look across cancers to see some potential molecular signatures for patterns that emerge, and maybe eventually end kind of this organ-specific treatment strategy. Here's the clear cell paper, which really moved us beyond VHL and clear cell. Everyone knows in the field that VHL is very commonly altered in sporadic clear cell kidney cancer. You lose 3P, and then you have the mutation in VHL. But the good pathologists looking at these tumors, about 80 to 90% of them have alterations in VHL, whether they're a somatic mutation or they're a hypermethylation of the promoter. But we moved to a new class of genes where they're chromatin remodeling genes, where they're tumor suppressors which actually are located on 3P, and they have a mutation. And those actually lead to a lot of different transcriptional changes due to histone remodeling. And then we also saw that kidney cancer and clear cell has a shift to Warburg. That's something which, we had a great introduction, that clear cell shifts its metabolism. These high-grade cancers do utilize something called reductive carboxylation, and they feed glutamine in for metabolism. But besides clear cell, there's been all these other landmark papers which have really done great characterization of different tumor types. But this is actually leads to a ridiculous amount of effort. Having being on the chromophobe and the papillary TCGA groups, I can see that it's amazing how all this takes place. There's 500 tumor samples in clear cell. If you look at how many base pairs, how many SNPs there are, how many transcripts, how much methylation is present. Study one cancer event in TCGA, it takes, you have almost 1 to the 10 times 20 to the 29th data points, and it really leads you to really, we need computer analysis to actually understand these events and really interpret it, but then you need someone who is a clinician who understands how to bring it back to the bedside and actually, make some of these things actually relevant to patients. And here's just a picture of Watson as, he's in the lead right now, and Ken, our former champion, is not doing so well. And the other potential, which I think is really exciting, is the idea of looking across cancer types. This just came out in Nature a couple months ago, and really they categorized some of the data available from TCGA. Just looking at, you can't publish a lot of the information yet. There's a moratorium, as they call, but the sequencing data is available, and you could actually publish with one particular platform. And they looked across all these cancers, and really they found that there were really 21 gene signatures, and there really pushes for the future of really targeting a certain transcriptional or mutational signature rather than a particular cancer. And here's just a little cartoon that said, good news, sir, your prostate cancer is curable, but it clusters like ovarian cancer. We're going to have to, you have to share a room with the woman on the gyn-onc ward. So that's maybe where we're moving at some point, looking across cancers. So how do we begin to go to kidney cancer and split things from clear cell, papillary based on morphology? Do you look at it by mutation or mutational signature? Do you look at it by copy number variation? Do you look at it mRNA gene expression? Or you do some type of integrated analysis? But one thing you need to ask, is this going to be better than our existing technology? Here's our existing technology. I know there's a lot of smart people in the room. Can anyone tell me which one of these is the VHL-associated clear cell tumor? You lose VHL, you have upregulation in the HIF, you have upregulation of the VEGF receptor pathway, giving you potential responsiveness to TKIs. Can anyone tell me which one is the clear cell, which VHL association? Okay, so these are patients that I've personally seen at the NCI. The one patient has Cowden syndrome. It's a loss of PTEN. There's a SDHC, which is another Krebs cycle enzyme, a germline mutation in succinate dehydrogenase C. Patients with tuberous sclerosis can get clear cell. HLRCC, which is another Krebs cycle enzyme, fumarate hydratase germline mutation, they can get clear cell. And a newly discovered one called BAP1, they also get a very high-grade cancer. That's another involved in chromatin remodeling as well. So I think we're getting better than just looking at something under light microscopy, but we need to start figuring out which tools are we going to use. And this is one slide from the TCGA clear cell where we actually, it's always been known that clear cell loses 3P, but there's all these other tumor suppressors beyond VHL which are right there. And they're mutated at a pretty high frequency, PBRM1, SETD2, BAP1, and or the 2 through 4th, both common, but you see things like mTOR. We know that the rapalogs, everolimus and temsirolimus are approved for kidney cancer and do lead to some stable disease. So, but we have a new class of genes which have now are trying to be targeted, but they are tumor suppressor genes that are involved in chromatin remodeling. It might be pretty hard to actually target those. But if we move, instead of just mutation assessment, we can actually use, looking at understanding the transcriptome in kidney cancer. On the top, this is a great project from Celeste Simon at UPenn where they actually clustered things into a clear cell A versus a clear cell B. And they actually have some overlap with Bill Kalin's work where there's a HIF1, HIF2 clear cell and there's a HIF2 predominant. And they seem to cluster into two different types. The TCGA validated this, but also said, instead of just making things 1 and 2, type A and B, we'll call it 1 through 4. And again, that one subclass actually had better outcomes. So maybe we can do this, and actually in some of the trials they're actually going to try to validate some of these mRNA subclassifications. How can we use this at Yale? This proposal we just sent to the PRC using an active surveillance protocol for patients with small renal masses, saying that in VHL we don't manage patients until they reach 3 centimeters. Well, what about sporadic VHL-associated clear cell? Can you actually biopsy them, assess other genetic events such as chromatin remodeling genes, potentially follow patients, knowing that they have extremely low rate of mutations, and then maybe correlate some of these potential biomarkers that have been studied in the TCGA and correlate them with growth rates. We know that there's ridiculous amount of overtreatment in prostate cancer. The same thing holds true in kidney cancer. In prostate cancer, you have men who are impotent and incontinent. You see that right away. In kidney cancer, things like CKD, hypertension, cardiovascular outcomes, they're seen more down the road. So one of my things is trying to avoid overtreatment, and we're just really trying to push the active surveillance forward, and then can we actually integrate a biomarker scheme for a ready standard of care approach, which is usually for elderly patients, but it also could be used for younger patients as well. Now, where we move into papillary. Papillary has been based on morphology. We said, there's one which looks basophilic with thin papillae, one looks more eosinophilic with thicker papillae. We know that there are 2 syndromes, and one syndrome has a MET mutation, and one syndrome has a fumarate hydratase mutation. And the field has tried to link these things together for several years, trying to kind of find some reason why there is a similarity. But if you look at the somatic mutation frequency, MET is really very rarely mutated in papillary type 1 tumors, and fumarate hydratase is not mutated at all in papillary type 2 tumors. So this is really kind of a myth that there is a link between these sporadic and hereditary syndromes in papillary kidney cancer. So it's much more complex than previously thought. But where I trained at the NCI, it was very unique because urologists ran a lot of the systemic therapy trials, and this was one that we participated, we ran, which was giving Avastin and Tarceva to patients with HLRCC kidney cancer, and we included patients with sporadic kidney cancer as well. In the lab, there was good preclinical evidence. If you lost the Krebs cycle and you needed to rely on glucose uptake, the Warburg effect, if you used Avastin, which blocked angiogenesis, Tarceva actually in the lab actually can block somewhat of glucose uptake. You can starve the cells of their glucose and they would die. And here's just looking at a clear cell line and an HLRCC line, just starving it of glucose for a day, the cells all died. So in this trial, initially, I saw some patients with HLRCC responding great. Other patients with sporadic ones actually didn't respond well. So what was actually describing this? And you can go back to the big data, and you can see how they actually cluster. If you look at expression profiles from HLRCC and papillary type 1 and type 2, HLRCC is in left field, and we're treating these patients the same way on a protocol with patients with papillary kidney cancer. And if you just pull out the genes responsible for glucose metabolism, you see on the right the ones that are in the red, those are HLRCC. They have completely different in terms of their glucose metabolism. So based on that, you could predict that if you had cell lines like we have here, if you starve the sporadic forms based on the gene expression profile and what would predict, you would expect that it would behave very differently. And HLRCC, the cell line, the cells mostly die without glucose within a day. And the sporadic ones, they don't care. They rely on glutamine. They rely on other types of carbon precursors. And then for papillary type 1 tumors, there's a big push to do MET inhibition based on, hereditary papillary renal cell is a condition which has only been described in 20 families that have a germline activating MET mutation. So we have drugs. This was a trial we ran at NCI with XL880. It's the Exelixis drug before they sold it to GSK. Now they have a new molecule, XL184, which many of you may be familiar with, which is also being studied in papillary kidney cancer. But really just saying morphologically, are we going to treat these patients with this drug because we say they look like a hereditary syndrome? We saw the overall response rate was actually pretty poor. It was only 13%. But if you actually looked at the molecular characterization, the patients who have activating MET mutations clearly are going to respond better. And in fact, they did, that the patients who had germline MET mutations, half of them had partial responses. And most of these patients were treated at the NCI, and every single patient that I saw with this ended up having at least some partial response, or were on their way to a partial response, but were taken off for toxicity. But just giving it to everyone, with just based on morphology is a little silly in my opinion. The papillary TCGA is underway. It was a little slow to accrue. Dr. Linehan at NCI is leading it, but Dr. Gerstein in the Bioinformatics department and myself are involved, and it should be a really exciting breakthrough in understudied kidney cancer. Just one slide on the chromophobe kidney tumors. That's the third most common. This is actually about to, we're putting it forward. The manuscript is finally going around. I'm actually the manuscript coordinator, and in papillary, in chromophobe, we have seen some patients have responses to mTOR inhibitors. But it's really hard to do a large study on patients with chromophobe kidney cancer because there's probably only 30 to 40 in the whole country a year that get that cancer. But we see that P53 and P10 are the most commonly associated alterations. We're doing a validation cohort with another 200 paraffin specimens, but that may be the rationale why some patients who actually respond to everolimus or temsirolimus. And you see actually chromophobe actually has a much lower mutational burden, but is characterized by widespread instability and loss of multiple chromosomes. So what are we doing with this at Yale to bring this information from the genomics era back to the bedside? Well, one, we have an exciting new kind of hereditary kidney cancer program collaborating with Alan Bale, Ellen Matloff, Jeff Sklar's in the back over there, and a great dermatologist, Christine Coe, and we're really trying to focus and get patients with early onset kidney cancer, bilateral multifocal kidney cancer, that they're going to be evaluated, be sequenced. If they go to surgery, we'll have the tissue. We can potentially characterize it. Cytogenetics program we're starting, so we can actually have biopsy specimens. We have nephrectomy specimens. It has prognostic methods of assessment. You can also look at certain different markers which have prognostic impact. And then FISH analysis, we have we have three different types of kidney tumors with translocations, which are really difficult to detect, but they occur in young individuals. And Jeff's team in the lab has just got a TFE3 probe, and it's a break-apart probe, and it'll be able to detect some of our patients, and we just found a couple tumors to study. And the active surveillance trial I told you, that's really standard of care, but we're really integrating some of those markers from the TCGA to kind of come up with maybe a predictive tool how we can really select who would be appropriate candidates so they can avoid unnecessary treatment. Oncocytomas are benign tumors which are copy number neutral, and they are very difficult to distinguish in biopsy from chromophobe tumors. So they actually have complex 1 mitochondrial mutations, another Warburg effect, and we're talking about either looking at markers of complex 1 loss or copy number variations, and Dr. Adenir, one of the pathologists, has put together a tissue microarray, and hopefully we'll move that forward. And then sarcomatoid transformation is one of my big areas of interest, and Rick Lifton has taken a couple samples that I had studied over the past couple of years, and we've actually done exome sequencing of the matched clear cell and the sarcomatoid region from the same tumor, 25 matched pairs, so we can understand the explosion that takes place on these tumors. Again, that hereditary kidney cancer program, there's been no guidelines for hereditary kidney cancer testing. You know, we know in breast cancer and colon cancer there are recommendations. Fortunately, we just had a paper accepted the JCO. We're looking at the NCI experience, and that's going to set the threshold of genetic testing at age 45 or less. But the guy I just saw in clinic today, 32 years old, had a large kidney tumor. Why is that guy getting kidney cancer? I don't know. He has no risk factors. That's the guy who needs to be interrogated. And then, you know, what Alan Bale has set up here is, you know, different from the classic, you know, traditional one gene at a time. This is a new technology which Ambry's really done, this panel-based testing. This is what Alan has here. This is state of the art, where they just basically, we have 15 genes in the hereditary panel for kidney cancer, but he looks at the other 28, 30,000 genes. You know, this is going to be really state of the art. These patients have this information, they go to the OR, we can make a cell line out of it, and really can characterize the genetic basis of their tumor. And then the cytogenetics, just one or two more slides. You know, there's a group of patients who have a germline translocation in 3 to 8 or 3 to 6. Penning Lee's lab is really helping us with solid tumor cytogenetics, and also will be helpful for germline analysis. And then this is what Dr. Sklar's lab is really helping us really trying to figure out. You know, it's only 1 or 2% of kidney tumors, but in patients less than age 30, this accounts for about 1 in 4 patients with cancer. And we did a partial nephrectomy on a 25-year-old guy last week, and it's running in the cytogenetics lab to look for translocation. And if it fails, we'll have a FISH probe to test. And again, prognostic, you know, for copy number variation, there's also other things. And again, oncocytomas, we remove way too many small tumors that are benign, and if we can biopsy these patients and actually use that information from the TCGA chromophore project, knowing that oncocytomas are copy number neutral, we can maybe not do unnecessary surgery on a lot of patients. So conclusions, you know, this genomic characterization is now taking place. We need thoughtful clinicians. You know, at Yale, there's so much resources here. We need to think of ideas to kind of bring this back to the bedside. And our kidney cancer program's expanding. Harriet Kluger, Mario Stoll are experts in the field. They've been great to work with. They're experts in kidney cancer. Now we have a urology department finally, which will be kind of equal partners and push this forward. So.
So as you can see from Brian, you may want to think about doing something to get a little more energy. Okay.
I'm a little hypomanic when you're talking about kidney cancer.
You're a little chill, you know. But that was a fantastic talk and really fabulous. Questions? Yes, Dr. Roberts.
Brian, so far as I know, with regard to treatment of metastatic kidney cancer, we have 2 agents for which we have a tremendous unmet need for predictive markers to response, IL-2 and the VEGF growth factor receptors like Sutent. Could you comment on where this field has, what's tried to do in terms of predictive markers?
So in terms of IL-2 response, there was UCLA, that's where I did my residency. They actually looked at carbonic anhydrase 9, which is a protein to basically, it's downstream of HIF, and they thought that it would basically predict for response to IL-2. And it turned out in the SELECT trial, which was 100 patients with led by David McDermott at Beth Israel, that it didn't really respond. It wasn't actually predictive. And I believe the reason why is, that CA9 was probably a marker of clear cell, and there were probably other, non-clear cell tumors in there which we know have worse response. And it was in a different era of pathologic classification. I think there was a lot of heterogeneity, and probably it just selected for the real clear cells. And now in the 21st century, when we understand really what clear cell is, I think it didn't pan out. But they were looking at other markers as well. B7H1, I think Dave McDermott had an abstract showing that it may show more immunogenicity of the tumor, potentially that. In terms of response to Sutent, I know Brian Reed and Bob Moser have looked at VEGF receptor SNPs and other things that may predict response. But in terms of biomarkers of response, actually with axitinib, if you have hypertension, some of these things, they're on-target effects, it actually might be predictive of response. Not necessarily prognostic, but predictive.
So, Brian, one question to clarify. You mentioned there were 28,000 genes. Now, Rick Lifton, last time he talked, said there were 23,000 genes in his whole exome. Are those 5,000 genes that we've discovered since Rick?
I just made those ones up.
Okay. So, but whether it's 23 or 28,
Yeah.
okay, some people have thought that maybe one of the reasons that certain tumors do well clinically, or may even be tumors that respond to immunotherapy, maybe some that have the hypermutated phenotype. Have you found anything like that in your Cancer Genome Atlas work that explains why some kidney cancers may respond to anti-PD-1 or even IL-2 based therapy?
I mean, unfortunately, like for the chromophobe specimens, all those patients live, so we can't do any correlations for the.
Unfortunately, they live.
No, I mean in terms of doing good prognostic kind of markers, but in clear cell in the TCGA, there's not much information in terms of clinical response. But there's an effort led by MSKCC and MD Anderson to look at the clinical information for those patients that went on to receive systemic therapy and maybe correlate it to response of things like IL-2 or PD-1. But it is a small subset of the clear cell 417 patients, so it's going to be much less. But James Shea at MSKCC and I think Feroz Tamboli and a couple of people from MD Anderson are looking specifically at trying to correlate like the hypermutated ones to actually response to some systemic therapies.
Harriet, last question.
Brian, can you comment on the role of systemic therapy in patients who've got relatively small tumors and we're trying to do nephron-sparing surgery on them?
So in terms of systemic therapy, in the immunotherapy era, there was in the IL-2 era, which was, not that IL-2, Harry and I have discussed, it's still a big role, but the primary tumor never really responded to systemic therapy. And if you look at waterfall plots of patients who were treated at the NCI, 52 patients with primary in place, they would have no partial responses and very small amount of change. In the new era of TKIs, the primary tumor is actually responsive, and there's about a 10% response maybe rate in patients who get TKIs up front. And Harry and I have actually put forward a proposal for XL184 to be given in the neoadjuvant phase for patients who have a large primary who would be a candidate for radical nephrectomy, but by doing a partial nephrectomy, they might have a benefit from nephron sparing, and we're hopeful to get that through. But this does have the potential that you're able to change your surgical approach, making a tumor which was maybe not amenable to a partial nephrectomy smaller, that it potentially could be ablated. For the most part, it's not that dramatic in the responses, but at least in a small phase 2 with axitinib, there was actually 9 of 19 patients had a partial response, and that's from MD Anderson.
So, Brian, I want to thank you. Now, the one unifying message is usually after this, I go downstairs to the candy store. Other people call it the drugstore. Go down to the candy store and I have a Snickers bar. But after listening to the two of you, I am not going to have a Snickers bar today. Keep those glucose levels down. Thank you both very much. Fabulous.